First, thank the meeting organizer to give me this opportunity to talk about our work on the live attenuated influenza vaccines. As you all know, the HA and NA surface glycoprotein protein of the flu virus keep mutating and cause antigenic drift. So the vaccine strain need to be updated every year. Here show you the all the vaccine strain components over the years in our live attenuated vaccines. So recently, the quadrivalent live attenuated vaccine has been approved, and it covers the H1N1, H3N2, and 2B strains. So just give you a brief overview of the live attenuated intranasally administered influenza vaccines. So for the seasonal ARV, so we have the trivalent ARV has been approved in the U.S. since 2003, and the recent approved in other countries. And the quadrivalent approved in U.S. in 2012 and Europe 2013. So besides this, we also developing pandemic live attenuated vaccines for different subtypes with pandemic potential. And we have complete phase 1 clinical trials for 8 strains. And this is the process we make the vaccine seed every season. So we use a powerful reverse genetic system to make the vaccine seed. So this is a 8 plasmid reverse genetics system. So the vaccine virus is a 6-2 reassortant, conferring the temperature-sensitive cold-adapted attenuation phenotype conferred by the Macedonia virus. And the HA and the NA genes are from the wild-type virus. And so every year before the WHO strain selection announcement, we have to make multiple SIG2 virus candidates from different wild-type virus. So we've gone through called the strain evaluation selection to select the best vaccine candidate. So the vaccine virus has to grow very well in embryonated chicken eggs and maintain the phenotype, and antigenically should be accurate, and also should grow efficiently in hosts and be immunogenic and keep the genetic and thermal stability, and also manufacturable. And I will give you some examples for the H1N1 and H3N2 strains, how we overcame some challenges and to make a high yield vaccine seed, and also improve the thermal stability of the vaccine. So here give you, so in 2009, when the H1N1 was caused pandemic in humans, we have to make the vaccine immediately in response to the pandemic. So, but when we first received the virus, the virus without any change, it cannot be rescued because the original virus is, has not been adapt well in cells and eggs. And when the virus is adapting in eggs, so there are single mutations around the receptor binding site make the virus can grow in eggs, but not as good as we want. And also there's this mutation lower the immunogenicity of this vaccine strain in ferret. So we cannot use these. So finally, after quick cell adaptation, we were able to get another substitutions in the HA gene, which can significantly improve the virus titer in eggs, and also has good immunogenicity and antigenicity, make it can be used in vaccine. So the live attenuated vaccine for H1N1 pandemic is the first vaccine shipped to the U.S. government. So you know the H1N1, the California-like strain has been introduced to human since 2009, become a seasonal strain. So we have to make these vaccine candidates every year. But unfortunately, most of the H1N1 strains cannot grow well in eggs, and of these, so from these candidates, you can see only 2 can grow well. So from these high grower, we were able to find other amino acid changes in the HA which can improve the virus titer. So these two different substitutions you can see can confer the growth of California beside the original one we found. And you know, recently the H1N1 strain keep evolving and form different genetic subgroups. Currently, so almost 100% of the circulating H1N1 strains are like 6B in the 6B subgroup. So you can see again, almost all of the strains we work on cannot go well in eggs. So we have to improve the titer. So you can see the strain we work on, the A. Bolivia strain 2013, when we introduced these mutations we found in the previous strains, it can improve the titer to meet the vaccine use. So again, the HA substitutions improve the viral titer, but doesn't affect the viral immunogenicity and antigenicity. So these can be used. So interestingly, you can see all these changes are changed to acidic amino acid residues in the HA head region. So we did some further analysis to see what stage it helped the virus growth in eggs and also in cells. So this is a kind of a low MOI infection data. You can see, so the acidic residue, a single residue can facilitate virus multi-cycle replication, especially in the virus release stage. So we hypothesize is the acidic residue change in the HA head region help the virus growth, possibly by facilitating virus, multi-cycle replication, and also these can be used for vaccine. And another thing is we found like the new H1N1 strain, like California 709 strain, is less stable than traditional seasonal H1N1 at a high temperature, like more vulnerable to high temperature. So we want to look at this issue by looking at the virus fusion, these activities, because we know the virus, the threshold of pH for the virus fusion correlates with the structural stability of the HA protein. So you know the fusion activation is important for virus replication and host adaptation. And so if the virus structure, if less stable, it will have higher fusion pH and the lower acidic and thermal stability. And also the fusion pH threshold is related to the host adaptation. So different hosts may have a different optimal fusion pH for flu virus. So we want to see if the California 709 strain has different fusion pH than other virus. So we compare these two H1N1 strain, which can, the Brisbane 1010 strain, which can grow well and it has better stability than California 09. Then when we establish a fusion assay, and you can see the California 09 has a pH fusion at 5.4, the threshold, and the Brisbane 1010 is 5.0. So that indicates California 09 is less acidic stable than Brisbane 1010. And we further map out which residue in the HA2 region is responsible for this difference. And it's interesting is, in the HA trimer, there's an interaction as a salt bridge exists if the HA2 region has a K at 47. But in the California 09, the HA2 at 47 is a E, which doesn't form acidic, the salt bridge to the HA1E21. So if in this case, like in the California strand, the fusion pH is 5.4. So if the Brisbane 10, the K mutated to E, the fusion pH is also 5.4. So the other way is if the California E changed to K, the pH became 5.0. So that indicates, so this residue is responsible for the fusion pH difference. So is this difference also correlate to the thermal stability? So we did a quick, the high temperature treatment for different strands. So you can see the California 09 strand did have lower stability. So the cutoff temperature for virus inactivation is 55 degrees. But for the other strand with K47, like Brisbane 1010, and the mutation of California changed back to K, it improved the thermal stability. And fortunately, the recent order H1N1 like evolved from E47 to K47, probably more adapted to human population. So all the new strands like Bolivia 2013, I just show, also has a good stability profile, similar to this one is similar to the Brisbane 1010 strand, the stabilized California version. So because of that, we aim to replace the 709 strand with Bolivia's 2013 to improve the thermal stability, or like more represent the recent H1N1 strands. Okay. So I will switch to the H3N2 story. So you know the H3N2 influenza virus also very hard to grow in both eggs or MDCK cells. It grows very poorly. And it also is the most frequently drifted subtype. So this picture just show you all the egg adaptation changes we saw in previous H3N2 strands of multiple years. So we from these years of work, we identify several amino acid change which are not good for the virus immunogenicity or antigenicity. So this is a summary of the historical data we found. So basically, it's quite consistent. Besides all these changes, these are the three main changes we found in different historical H3N2 viruses. For example, the G186V change, which is required for all these changes, is required for virus to grow well in eggs, but we have to evaluate these changes for the impact on the immunogenicity and antigenicity. So we usually do a ferret study to make ferret antisera and test in the HI against the different virus. So you can see the 160 H186 change, which is okay for the immunogenicity, also cross well to the cell-derived virus, which should be the reference virus for detected antigenicity. And the L194P change, which is, you can see, significantly lower the immunogenicity, so the homologous HAI titer sometimes is non-detectable. And the H183L change, which is good, so it has good immunogenicity and good antigenicity. And if the 156 change, which is really around the receptor binding site, so that affects the vaccine antigenicity, but not the immunogenicity. So because it doesn't cross well to the cell-derived virus. So recently also CDC began to use the cell-derived virus as a reference strain, not the egg-derived virus as a reference strain. So we noticed this a long time ago is if you use egg-derived virus as a reference strain, it won't accurately predict the real antigenicity. For example, if you use the 194P chain P virus as a reference strain, the GU86V virus cannot cross well. So that's our conclusion. And again, similar thing happens for the recent strains. So with, for example, this New York strain, with this only single amino acid change, you can see the different pattern of immunogenicity and antigenicity. And again, the California, the Texas strain, which is the previous, the recent vaccine strain, it show a good immunogenicity, but has about a fourfold reduction in the HI2 the cell virus, which CDC also noticed this difference. It's not really ideal for the vaccine use. And for the 194P change also identified, but the H183 change, so this one we identify and think this could be a good vaccine candidate. So this year, the H3N2 drifted in 2014 and 2015 season, and we so the current, the Texas-like vaccine strain cannot cross well to the recent H3N2 strains. So this because of sometimes the vaccine strain cannot predict the real the circulating strains. And the 2A, 3C, 2A, and the 3C, 3A strains, or the majority circulating strains, which has some drift to the Texas vaccine strain. And we need to, for this year, we need to prepare a new H3N2 vaccine candidate. And this year, we found some new adaptation changes in this new virus. And the Switzerland now is selected as a vaccine strain for the 2015 to 2016 season. But this one, again, similar to the previous virus, has some reduction. And but all the strains has some unexpected change in the HA, especially the glycosylation site change. So in the next seed, our goal is to generate the 3C2A candidates vaccine virus, because this one is a majority in North America compared to the 3A virus. So we have to generate this 3C2A candidate vaccine virus that grow well, but without affecting the viral immunogenicity and antigenicity. So that's our goal. So this is a summary. So for the H1N1 strains, we improved the virus yield in X by identifying the critical residues in the HA1 head region. So we were also able to identify a critical residue in the HA2 stalk region, which affects the viral thermal stability. And for H3N2, So we identify multiple egg adaptation changes that affect the viral immunogenicity and antigenicity. And the new drifting of the H3N2 virus poses more challenge to select the vaccine candidate to grow well in eggs and maintain the immunogenicity and antigenicity. But overall, so this live attenuated vaccine has been proved for many years that it's effective in children for matched and mismatched strains. And we think the virology technique will help us to get the best vaccine candidates. And this is an acknowledgement. So in our California site, we have been delivered the vaccine seed from 2003 to 2014, almost 10 years, successfully deliver the vaccine seed every year. And but the company changed the strategy, so the California site, the function transferred to the Liverpool, the UK manufacture site. So this is a new team they built in Liverpool, UK. So hope this vaccine can be delivered successfully every year. Thank you for your attention.
So thank you for the great talk. So now it's open for discussion on it, please.
Thank you. So you're, for the reverse genetics, you're starting with a DNA sequence. Mm-hmm. How do you choose that sequence to start with? I mean, there's different...
You mean the HNAG, right?
Yeah.
Uh-huh. So we usually receive white-type virus, right, from CDC. So white-type virus may contain multiple sequence populations in the white-type virus. So we clone the gene from the white-type virus, like PCR, and clone the gene to the vector. But we will get multiple different variants from this white-type population. So then we test in, for the growth, for the immunogenicity in ferrets. So that's contain multiple evaluation to select the best variant, which is a single sequence population.
So it's purely empirical. You don't have like an algorithm to help you select which of the sequences in that population.
You mean?
Like a computer, almost like a computer.
No,
program.
we didn't do computer modeling.
And have you ever tried just growing it in the cells, not switching over to the eggs, but just going all the way to
But our
vaccine?
vaccine platform is egg-based, so we still cannot use cell too. But even the cells also, the natural influenza virus cannot grow well in MDCK cells.
Are there any other questions on the floor? Yes, please.
Very simple question. I saw in several places about immunogenicity and antigenicity. Mm-hmm. So what is the difference between immunogenicity and antigenicity?
Okay, yeah, this is a good question. Immunogenicity, we usually, so we use ferret study to evaluate both immunogenicity and antigenicity. How to define immunogenicity is how the antibody level elicited by a vaccine strain. So you inoculate vaccine strain to ferrets and collect the ferret antisera and see how it cross-react well to your homologous virus. So see the HAI titer to the homologous virus. That's called immunogenicity. So the level of antibody response. But antigenicity should be how it reacts to the real, to the real cell-derived, the real circulating vaccine strain. So you do the cross-reactivity to different circulating strains, also to the real reference strain. So we call the antigenic reference strain. Now should be the cell-derived virus, should be the real clinical isolate. So how the antisera can cross well, not only to the homologous, the vaccine virus, but also to the circulating white-type virus. That's called antigenicity. So the difference should be less than fourfold. That's the certificate, I mean, the CDC's criteria.
Are there any questions? Are there any particular reasons, for the H1, it's this, the mutation is somewhere around 120, 5 or 127, and H3 for the 190, 180. Are there any particular reasons for this sort of structurally in the HA?
You mean the site?
Yeah.
Yeah, so I think a different sub has slightly different mutation to help the virus growth. But the residue I show in H1 is the H1 numbering.
Yeah.
For the H3 is the H3 numbering.
Right.
So it's a slightly different, but there's some common, like say 225 change, which is according to H3 numbering. So for the H1 numbering is 222. So this residue is important for both. But some of the residue is specific for H3, some of them specific for H1.
Thank you.
Okay.